Good afternoon. This is Pat Steeg coming to you remotely from the NCI in Bethesda, Maryland, and we're going to go over 5 different very interesting posters. The first 2 deal with trastuzumab deruxtecan, and we'll take them more or less one after the other and see what they're telling us. The questions that I think we can talk about is, can preclinical data inform the brain metastasis trials in terms of efficacy, in terms of activity in HER2 low brain mets, and updates on the clinical activity of this ADC for brain mets. So a little background. I suspect we all know this about trastuzumab deruxtecan. It's a HER2 monoclonal antibody linked to a topoisomerase inhibitor via a cleavable linker. Approved in adult patients, unresectable or metastatic HER2 positive breast cancer who've had 2 or more prior HER2 treatments in the metastatic setting. In the Destiny trial, patients were excluded if they had untreated or symptomatic brain mets. So we're just starting to learn about its activity in the CNS. After binding to HER2 on cells, T-DXd is internalized and the linker's cleaved by the lysosomal enzymes. And once it's released, the deruxtecan is very permeable of membranes. So it can bind to and inhibit topoisomerase DNA complexes, and it can move from cell to cell, and that's an important point. It achieves a higher drug-to-antibody ratios compared to the other ADCs that are out, including TDM1. And as I mentioned, it demonstrates a bystander killing effect. So the DXd, once it's cleaved, can move to neighboring HER2 expressing cells or adjacent tumor cells that are negative for HER2 and kill them. So it's a very interesting compound. I made a quick guide to preclinical model systems for brain metastases, and we'll see 2 of them here. PDXs, patient-derived xenografts, have strengths and weaknesses. So here you're taking human tumor tissue and you're implanting it into the brain. And so it is human tumor tissue. This is a huge strength, although it may go through rounds of culture or in vivo growth in animals to start the experiment. And you see more heterogeneity and molecular and pathologic alterations in these different PDXs. The weaknesses is, how does this tissue connect to the all-important brain vasculature? So does it set up a true blood tumor barrier, and does that affect drug efficacy? Many people use intracardiac injections of brain-tropic cell lines. So that's the left ventricle of the heart. The tumor cells go everywhere in the body, and if they're brain-tropic, they will make hematogenous metastases in the brain that have a real blood tumor barrier. The weakness, it's an old cell line. We're using established cell lines. Occasionally, you'll see brain metastases from an orthotopic tumor, mammary fat pad injection. It's the real deal. It is incredibly slow, incredibly inefficient, and that's why you don't see much of it. So, what the authors have done for trastuzumab deruxtecan is the PDX model. So they were implanted, and then the mice were treated with T-DXd, and you're looking at growth by bioluminescent imaging, and you're looking at survival. In each case, the red line is T-DXd, and you can see that it extended survival. Here is a HER3 positive, 2 HER3 positive PDXs, 1 ER negative, 1 ER positive. And you can also see that the imaging, you get lots of bright red in the control and not so in the treated, and the same here. What's interesting, not much change in the tumors in Ki67 proliferation, but there is an increase in apoptosis. This looks like none to some as opposed to a high rate, but there it is. Here are two more models expanding. So this is low HER2, IH2 positive FISH negative. And again, you see the same benefit. And here is a TDM1 resistant model, same benefit. So what do we have? We have widespread activity. My notes are in blue here, including HER2 low and TDM1 resistant. I'd love to know if this, what was used was a clinically achievable dose and schedule. There are some experiments where there's a ton of injections and others less. I'd love to see data on drug distribution to the brain lesions. And I'd love to know more about the PDX model. Is it just a tumor that grows where it's implanted, or do these cells invade out and produce micromets? Just so we learn the model. Okay, this group also looked at data from patients they had treated with TDXD, and they have 17 patients who had CNS lesions and looked at the response, the cycles of treatment, and why they stopped. And here's the data, the swimmer plot, at least, of the data. And I'm going to give you an analysis that I made of it. So 10 of 17 patients in the CNS had a partial response. This is great. Of these, 6 of the 10 were patients that had systemic stable disease. 4 of the 10 had progressive disease systemically. And only 4 of the 10 patients who got a PR had baseline stable disease in the brain met. So this is active in a setting of uncontrolled systemic disease and active brain mets. I think that's very, very interesting. Low numbers yet, but wonderful news. Okay, and we're going to move to the second abstract, and this is TDXD in patients with HER2 positive or low advanced breast cancer and central nervous system involvement. And this is preliminary results from the DEBRA phase 2 studies. They're going to talk about 2 of the cohorts, A and C. This is my cheat sheet shown below. Cohort A is just HER2 positive advanced breast cancer with non-progressing brain mets. So they've been treated with radiotherapy or surgery. Cohort C is progressing brain mets after local treatment. They'd all had a prior HER2 therapy, a taxane. They got the standard dose. These are small numbers of patients, 8 to 9, and the follow-up is very short, 5 to 6 months. The primary endpoint is 16-week progression-free survival in cohort A, and they met it, which is unsurprising. I imagine that's not too different from historical estimates for that short a time period. The CNS overall response rate in cohort C is very interesting. It's a 55% partial response. So here is the enrollment data from the 2 cohorts. Here you have cohort 1, cohort 3. These are highly HER2 positive, so you have mostly 3+ disease. These are highly ER positive, 87% in one cohort, 78% in the other. So this is not a cohort that is indicative of all HER2 positive disease. This is mostly the luminal B type. These are also patients without widespread metastatic disease. 50% or so, over 50%, either had metastatic disease to 1 or 2 other sites. It's a cohort where many of the patients received whole brain radiation therapy, which we tend to delay more. And so this is a very characteristic cohort. Okay, what happened? Efficacy in cohort 1, as we said, 7 of 8 patients with stable brain mets at the baseline were alive. and without progressive disease, so they got to their cutoff point. This is unsurprising. Table 2 is the cohort 3. These are the active brain mets, and there was a 44% partial response and stable disease that lasted different amounts of time. Here are the adverse events, which for TDXD we need to start taking a good look at. And you see any grade or grade 3 to 4, and there is neutropenia, there is fatigue. I'm interested, I would like to hear the author's comment on that first footnote where they say 2 patients died of non-treatment related adverse events. One of them was an intraventricular hemorrhage. I'd like to know how that was non-treatment related. If there was disease in the ventricles, leptomeningeal disease, or what was going on there. Okay, small cohorts, very interesting data. The study centers on ER-positive, HER2-positive disease with limited follow-up. So we're going to need larger studies to more fully examine efficacy. As I said, I'd like a discussion of that adverse event. What should the primary and secondary endpoints be in these trials? I'd love to hear the patient's thoughts of what is a meaningful advance. Okay, we're now moving to the third poster, and this is the REGIST-EM study. It's a non-interventional cohort study, and it provides prospective data from around 1,800 advanced breast cancer patients. And what they're going to talk about is their data on 218 patients that had brain mets that were included. These patients are 97% Caucasian, mostly postmenopausal, and the distribution is only given in broad terms. So luminal, ER positive, 41%. HER2, regardless of ER status, 35%, triple negative, 19%. 80 of the patients, or 37%, had brain mets at the diagnosis of advanced breast cancer, and in 17 of them, the brain met was the only site of relapse. Now, there's some really interesting data here that is new to my knowledge. One, overall survival from the brain met diagnosis was 13 or 4 months in patients who had brain mets at or after ABC diagnosis. In other words, concurrent brain mets correlated with longer overall survival. I've not heard that. The Cox regression time to brain met development, again, surprised me. De novo metastatic breast cancer associated with a longer time to brain met development with a good hazard ratio. The triple negative subtype was associated with a shorter time, and that is consistent with all the literature, and everything else was insignificant. So is this what other people have seen? I'd love to know if other people have seen those trends in survival. And of course, the question is why? Why is overall survival from brain met diagnosis longer if brain mets were present at the initial metastatic breast cancer diagnosis? Why is de novo MBC associated with a longer time to brain met development when it's de novo? So the questions I would have is death due to systemic disease or brain mets. You know, as this cohort matures, it would be wonderful if the authors could fill in some of this data. The de novo brain met patients at ABC diagnosis typically had early breast cancer, 81% of them. So is there something in early therapy that is failing to protect against brain mets? And should we be thinking about that? Is the close timing of stereotactic radiation, gamma knife, and chemo in those patients that had de novo brain mets at metastatic breast cancer beneficial? Or is it just that by the time brain mets have developed after the metastatic breast cancer diagnosis, tumor cells are incredibly chemo resistant? And I'd like to know to what extent these trends are driven by the preponderance of luminal brain mets in this cohort. Did they get brain mets when they were on hormonal therapy or chemotherapy? And I'd love to see the data broken out into other subtypes. Okay, moving on, we have another interesting poster that is an assessment of the genomic landscape of brain metastases. Very interesting. This group had 822 brain metastases to look at, okay, compared to almost 12,000 primary breast cancers and 15,000 metastases that were not to the brain. So this is an amazing data set. And they looked at genome effects and they looked at protein expression by IHC. So here's a summary, my cheat sheet of the genomic alteration frequencies, and you can see the brain mets right here. If they look for loss of heterozygosity and call that HRD, setting the usual 16% genome wide, they're saying that 52% of these samples had homologous repair deficiency. If they looked at mutation burden, called it high here, it was 16%. Microsatellite instability is very low. And PD-L1 genomic alteration was seen in almost 40%. I think this is the more interesting of the data tables, and this shows some of the protein patterns. And I've put some blue arrows here that I want to point out to you. And what this is positive PD-L1 expression in the brain met. This is the 1% cutoff that's used in melanoma trials. Look at this. This is 23% in the ER positives, 27% ER positive, HER2 positive, 57% ER negative, HER2 positive, and 48% in the triple negatives. These, to my mind, the triple negative is not a surprising number, but all the rest of them, to my knowledge, are surprising numbers. This just shows you mutations and copy numbers. And very quickly, again, for discussion, how does this compare with data from other institutions? I think the surprising finding here is that PD-L1 looks pretty high in brain metastases in multiple subtypes. How high is high enough to incorporate into trial? We don't do brain met biopsies, but should we ever be considering them? They saw a nice change in cadherin-1, and I'm unsure. I'd like to hear a discussion of their support for using PARP inhibitors. There are better tests out than just LOH for homologous repair, and even if there is a homologous repair deficiency, is PARP the only target there? And many PARP inhibitors are poorly brain permeable. Moving on to the last, this is a preclinical paper where investigators took models of breast cancer brain metastases, and they isolated latent human tumor cells from the mouse brain. And so they're looking at dormancy, and show that these latent tumor cells are resistant to many therapeutics, but that they overexpress the AXL gene, and that could be interesting. So let's look at the data. So basically, this is the orthotopic injection. This is the real deal, a primary tumor, and they get some with brain mets, and they get others that don't have brain mets, but there's some cells in the brain, and these are called the latent cells. And what they show is that these latent cells in this middle panel are resistant to lapatinib, neratinib, tucatinib. What they also show here by Western blot is that these latent cells, as well as the frankly metastatic cells, overexpress AXL. And if you look at 5 sets of matched primary tumors and brain mets, these are now human, I believe. you see a increase in AXL expression. If they knock down AXL in the tumor cells, so here's the knockdown, and do the same injection. Here is the brain photon flux. That's all they're showing. They show it here in the animal and here in the resected brains where the skull isn't getting in the way of the imaging. And you can see a nice knockdown. What I think is much more interesting here is the data looking at these cells, the latent cells now that they have isolated are sensitive to a number of therapeutics in 2 model systems. Here is where the data gets a little interesting, where they've used bemcentinib, which is an AXL inhibitor. I don't know the dose and schedule, but here is the imaging again, and it's not, it may be significant, but it sure isn't a home run here. Ex vivo, it looks a little bit better, so I'd like to hear more about this. AXL is a widely known gene in metastasis and in brain mets, so this is very confirmatory. And what I'd really like to know is the efficacy of bemcentinib, the dose and schedule. Was this in a clinically achievable? And what would be the data if dosing was started later than day 2 post-injection? So this is very early and continuous treatment to prevent, and a clinical update on this drug. Thank you very much. I look forward to the discussion.